guys, I'm back again with some A2 biology. We're going to be looking at aerobic respiration, as it says up there. There's two types of respiration. There's anaerobic and aerobic, and anaerobic is without any oxygen, and aerobic respiration is with the presence of oxygen. Now, ironically, in aerobic respiration, the only part of it that actually uses any type of oxygen is this one here, oxidative phosphorylation, which is the last step. But anywho, it still uses oxygen, so therefore it's aerobic. Respiration as a whole is, the process whereby energy in organic complex molecules is used to produce ATP. And ATP is the universal energy currency, and we need this energy to drive our metabolic processes, which essentially keeps us alive. So ATP looks like this molecule here. It's got a ribose sugar, nucleotide, and 3 phosphate groups. So ATP is a phosphorylated nucleotide. And because of the 3 phosphate groups, it is called adenosine triphosphate. Yeah? And respiration occurs in the mitochondria, so the matrix of the mitochondria. We've got 4 steps of aerobic respiration. We've got glycolysis, the link reaction, the Krebs cycle, and oxidative phosphorylation. Yeah. So let's go into the first step, glycolysis. Now, don't be fazed by all of this writing. It's really not that bad. Okay, so we've got glucose, like it says in our equation here. Glucose plus 6 moles of oxygen gives us 6 moles of carbon dioxide and water. If you notice here as well, it's a reversible reaction. Carbon dioxide and H2O will give us glucose and oxygen, which is the equation of photosynthesis, which I'll go into another time. So back to glycolysis. Here we've got our glucose, which is our respiratory substrate here. Glucose has 6 carbon atoms. Okay, now the first step is glucose is phosphorylated. This is done by taking a phosphate group from our adenosine triphosphate, our ATP. So taking a phosphate off that, giving us adenosine diphosphate, which just means it's got 2 phosphate groups. So this here is phosphorylation of glucose, giving us 6 glucose phosphate. This is then isomerized into another 6 carbon compound here. So that is 6 fructose phosphate. So that's got 6 carbons as well. And then if you notice, our next product is 1,6-fructose biphosphate. This is supposed to say biphosphate. This means that there's 2 phosphate molecules, but up here we've only got 1. So therefore 6-fructose phosphate has been phosphorylated. So ATP then phosphorylates this, giving us ADP and giving us 1,6-fructose biphosphate. This is then split to give us 2 triose phosphates. Triose means 3. So this means we've got 3 carbons, which makes sense because here we've got 6 carbons. So the next step, after we've made our 2 triose phosphate molecules, we have to take away one of these phosphates and take away some hydrogens. Now, how we do this is we then begin to phosphorylate adenosine diphosphate. So ADP makes ATP. So now this is then when we start making some of this ATP. We make some ATP. But if we take away some phosphate, we also have to take away some hydrogens. So we take a coenzyme, which is NAD, and then we hydrogenate this by reducing it with a hydrogen atom from our triphosphate. And we do this for this molecule as well. Okay, and this gives us an intermediate compound that we do not need to know, thankfully. And then finally, our intermediate product is phosphorylated again by ADP to give us more ATP, hooray. So then we then have pyruvate. Now these are all still 3-carbon compounds. And pyruvate is our end product of our first stage of respiration, pyruvate. So if you notice, we haven't really taken away any carbon. We still have 6, and they were just split to give us 2 triose phosphates. If these were combined, we would still have 6 carbons, same as glucose. So nothing was decarboxylated in here. So now let's do a summary of what was made. We made 1, 2, 3, 4 molecules of ATP. So we made 4 molecules of ATP. However, we used 2 to phosphorylate our glucose and our 6-fructose phosphate. So therefore we used 2. And because of this, our net production of ATP is 2 ATP. And then we made 2 molecules of reduced NAD. And this is glycolysis done. If you keep going over this, you'll realize that it's not as interesting, it's not as hard, as it looks. It's actually quite easy. Okay, so this glycolysis. Now, these our ATP and our NADH will be kept in the matrix for other things. So NADH especially will be used later on, and ATP will be used for metabolic processes. So then after this, we go into our link reaction. Now this here is our link reaction. So we started with our pyruvate that we made from glucose, from glycolysis, 3-carbon atomic compound here. Now what happens here is we make acetyl coenzyme A. So acetyl coenzyme A is a 2-carbon compound, so we must decarboxylate pyruvate, so we lose carbon dioxide to give us 2 compounds. But if we lose a carbon, we also have to lose hydrogens, so we dehydrogenate pyruvate as well, meaning we reduce some more of our coenzyme NAD to give us reduced NADH. This gives us acetyl coenzyme A. Now, if you see here, we've got acetate. This is also a 2-carbon compound. But here, we lose our coenzyme A to just give us acetate. This coenzyme then goes back to pick up some more acetyl. So here we go. And this acetate, this little bit here is our link reaction. All that really happens is, pyruvate is decarboxylated and dehydrogenated. Coenzyme A leaves, gives us acetate. And then we come into our Krebs cycle here. So this acetate that we've made in our link reaction joins with this 4-carbon molecule, oxaloacetate, to give us citrate. So if 2 carbons and 4 carbons join together, we get a 6-carbon compound. And this is citrate. Now, if you look here, it says we've got a 5-carbon compound. So as we knew from pyruvate, we have to decarboxylate citrate to give us 5 carbons. So we lose carbon dioxide. And if we lose carbon dioxide, we have to dehydrogenate to lose the hydrogens that were attached to our carbon. So we reduce some NAD. So we produce NADH. Again, we go from a 5-carbon compound to a 4-carbon compound. So again, we decarboxylate and essentially dehydrogenate. Here we go. And we lose some NADH as well. Now, we go from a 4-carbon compound to a 4-carbon compound again. So we're not decarboxylating. So here, we make some ATP finally. So this is where we start doing some phosphorylation. From this 4-carbon compound to this 4-carbon compound, we make a different reduced coenzyme, which is FADH, which for the purposes of what we're learning now is the same as NADH. So FAD is reduced, takes a hydrogen, and this 4-carbon compound regenerates oxaloacetate by dehydrogenating it and giving us reduced NADH. And that's it. That's the end of our Krebs cycle. Looks a bit difficult. Again, it's just a thing of memory. So how many ATP did we make? Well, we didn't make any in the link reaction. We made 1 molecule here. But for every pyruvate molecule, you have 2 cycles, 2 going on here. So if we made 1 molecule, that means overall we've made 2 molecules of ATP. With NAD, we've made 1 in the link reaction, and we've made 1, 2, 3. So we made 1, 2, 3, 4, giving us 8 molecules of NADH, sorry. Yeah. So our FADH, we've made 1, so we make 2 molecules of FADH, and carbon dioxide, 1 here, 1 here, and 1 here, so we made 6 molecules of CO2. And that's our Krebs cycle done. So that's it. And then we go into our final stage, which, if you remember, was oxidative phosphorylation. And this was the most difficult part for me. If I can just find that sheet. Okay, here we go. Here is our mitochondrial matrix. Now this space here is our matrix. This is our intermembrane matrix. And here is our intermembrane space. So here's our matrix and here is our membrane. If you can see, this is our biphospholipid layer with our enzymes, with our electron carriers. Now, yeah, these blocks here are electron carriers. So, and this molecule here is called an ATP synthase enzyme. And by the name, you can sort of deduce what it does. So we get our reduced NAD here from our, from our glycolysis, from our link reaction, and from our Krebs cycle. And these are all being stored in the matrix. And we get an accumulation of them. This then dissociates to give us the NAD we had, and 2 electrons and 2 hydrogen atoms. And this is also a reversible reaction. So if we had 2 NADs, 2 electrons, and 2 hydrogen atoms, we would also make reduced NADH. So this dissociates. Now when this dissociates, NAD goes off back into the mitochondria to pick up some more hydrogen from whether it be glycolysis, the link reaction, or the Krebs cycle. And then we use the electrons and our hydrogen atoms. Now these electron carriers do exactly what they say they do. They carry these electrons. It's called an electron transport chain. So this electron here goes into this electron carrier here, and is transported along these different electron carriers. Now, the energy that these electrons have pumps these hydrogen atoms through these electron carriers into the intermembrane space. So, hydrogen atoms are just being pumped into the intermembrane space. The pumping eventually as these electrons pass through the electrons and pump these hydrogen atoms, the concentration of hydrogen atoms in the intermembrane space will be higher than the concentration in the matrix. So that means that these have to be actively transported, and that's where the energy of the energy of the electrons does. Otherwise, the hydrogen atoms that are accumulating here, that is creating a proton gradient, will diffuse back through these electron carriers into the matrix where we don't want them. So they go through these electron carriers and create a proton gradient here in the intermembrane space. Now, the flow is called a proton flow force, and the movement of these hydrogen ions or protons, is called chemiosmosis. So everything I just described here, the movement of these hydrogen atoms that are hydrogen ions which are creating a proton gradient through these electron carriers, is called chemiosmosis. So, and it creates an electrochemical gradient, because hydrogen atoms, accumulation of hydrogen ions, creates a pH gradient. So these are all things you should keep in mind, but just for the understanding of what we're doing now, all you need to know is that these hydrogen ions are going to be pumped into this intermembrane space here. Now, as this happens, because these are being pumped into the electrons, into the intermembrane space, they then flow into what we call an ATP synthase enzyme, and this is the key to us making loads of ATP, because they flow in, and the energy that these hydrogen ions have drives the rotation of this ATP synthase enzyme, and it rotates, and as it rotates, it joins ADP, adenosine diphosphate, to an inorganic phosphate group to give us ATP, what we've been looking to do this whole time. So all these hydrogen ions are being pumped here so that they can just diffuse through the ATP synthase to make ADP. Now, our FAD that we also made from our Krebs cycle also dissociates, giving us two hydrogens, giving us hydrogen and some more electrons that flow through this electron transport chain, pumping these hydrogen ions across the into the intermembrane space. And these hydrogen ions then join with an O2 molecule, which makes this aerobic respiration, to give us H2O. So that's what is happening here. It joins with these hydrogens from FAD, join with an O2 to give us H2O. And if you look in our equation, if I can find it anywhere, if you look in our glucose in our respiration equation, you will see that glucose and oxygen gave us carbon dioxide and H2O and ATP, obviously. So here's our H2O being made, and our carbon dioxide was being made from our Krebs cycle. So here's our H2O, and all of these hydrogen ions then give us a total production of like 26 ATP molecules. So the whole of respiration and the whole of everything that I've showed you, glycolysis, the Krebs cycle, the link reaction, we make a total of 30 ATP molecules. And we have made this using only one molecule of glucose. So here we go. I've got like about 1 minute to finish this off. So if you've got any questions, just put it at the bottom of the video, and I'll be more than happy to go over this in more detail if there's specific parts of this that you don't understand. But yeah, have a good day, and yeah.